All right, so let us continue with the IGCSE Biology 0610. We now look at chapter 5, which is enzymes. So this is for the exams in the year 2026, 2027, and 2028. Now, that we are looking at chapter 5, it means that we've done chapter 1, chapter 2, chapter 3, and chapter 4. At this point, you should have gone through all those lessons, including exam past papers, that is for multiple choice. So if you've not done so, make sure that you go through before you come to this one. Okay, so from here, also at the end, we are going to answer exam questions. So I urge you to stay up to the end. All right, and do not forget to subscribe to this YouTube channel, share with others. You might just help someone. So if you're going through this lesson a day before an exam, you count yourself lucky because you're going to have all the information that you need as we prepare for exams. Otherwise, we can continue. So now, what do you need to know about enzymes? So enzymes are very important, okay? They play a role in controlling the metabolic reactions that occurs in our body. So a metabolic reaction, we're just looking at the sum of all chemical reactions that take place in the body, can involve breaking down or building. But that is not our thing. So a metabolic reaction is just a sum of chemical reactions that take place in our body or in living organism, let me say. Now, these metabolic reactions, they can be slow without enzymes. So enzymes are needed just to speed up the metabolic reactions or chemical reactions that take place. And for that reason, enzymes are known as catalysts. Now, let us understand a catalyst. So we can start now. So what is a catalyst? So a catalyst is just a substance that increases the rate of chemical reaction and is not changed by the reaction. So it just increases the rate of chemical reaction. So there are two things that you need to understand from the term a catalyst. One is speed up rate of chemical reaction and it remain unchanged. And that is exactly what an enzyme do. Okay, so now, since we said an enzyme is simply speed up rate of chemical reaction, but you should know also that this enzyme is made in the body of living organisms, and for that is known as a biological catalyst. So an enzyme is not any other catalyst, but it's the biological catalyst, and this is because it's made in living cells. Now, what is really an enzyme? What is an enzyme? So again, you need to understand the definition of an enzyme. Very important. As we start looking at questions, you're going to understand the importance of this. So there are two things or three things that you need to know about an enzyme. So an enzyme, or enzymes rather, are proteins, or an enzyme is a protein. So one thing you should know that it's made up of protein, and if it is made up of protein, then also we can say it is made up of nitrogen, because proteins are made up of amino acid and amino acid nitrogen. There's a reason why I'm explaining this, because there's a question at some point, which of the following, let's say it's true about an enzyme, is made up of proteins, made up of protein, then made up of nitrogen. So all that. So very important that you understand that an enzyme is made up of protein, but very important to know also what the protein is made of. What is the other thing that you need to understand about enzyme? Is speed up rate of metabolic reactions. So there are reactions that are slow, so they need enzyme. Okay, so it speed up rate of metabolic reaction. Aside from that, it remain unchanged. So at the end of a reaction, the enzyme remain unchanged. What does it mean it remain unchanged? It remains the same, and for that reason it can be reused. Okay, so the enzyme can be reused over and over again. So that is what you need to understand up to this point. And I guess you've taken or you've picked some point up to this point. Just from here, from the start, already exam questions, we are going to find exam questions. So I urge you to pay particular attention even as we continue. Let us look at the role of an enzyme in metabolic reactions or in living organisms. So the role of an enzyme in living organism is simply to speed up metabolic reactions. It's simply to speed up metabolic reactions. Now, very important to understand also that without enzymes, these metabolic reactions will be very slow. Okay? So without enzymes, metabolic reactions will be very slow. And these metabolic reactions are the ones that powers life. So without enzymes, actually, life would not be, living organism wouldn't exist. Okay, so without enzymes, living organisms, or I mean without enzymes, reactions would be very slow. Okay, now these enzymes maintain the rate of the reaction rates of all metabolic reactions that are necessary to what? Sustain life. I think I touched on that part already. So enzymes are very important where life processes are involved. Okay, so very important that you understand this part. Let us look at the importance of enzymes. Like I said, enzymes are very important. They speed up rate of metabolic reactions. Some of the examples of metabolic reactions that we can talk of is digestion. So you know that digestion, when we eat food, digestion takes place, and that is how we obtain body requirement or nutrients that are needed for respiration in the case of glucose. So if this was slow, surely organisms would not survive. So enzymes are very key. So digestion, and without digestive enzymes, animal would not be able to break down molecules quickly enough to provide energy and nutrients and they need to survive. So there will be no survival without enzymes. So very important that you understand that. All right, let us now turn our attention and look at the enzyme action. How does enzyme work, or the action of enzyme? Now, to understand this part, we need to understand few of the terms here, and one of the terms that we are going to look at is a substrate. Now, you cannot talk of an enzyme without mentioning or talking about the substrate, because an enzyme and substrate, actually these go together. So what is a substrate? So a substrate is simply the substance which an enzyme acts on. Very important. The substrate rather is just the substance which an enzyme acts on. Then when you talk of the products, we are just looking at molecules produced when an enzyme acts on the what? On the substrate. So very important. Okay. Now looking at the parts of an enzyme here, just looking at what is happening here, very important that we understand the parts of an enzyme. So the enzyme is this one at the bottom that you are able to see. Now this enzyme, it has this part and also the active site, which is the area of interest. So this opening part of an enzyme here is called the active site. Now what is the importance of active site? The active site, that is where the substrate bind. As you can see from this diagram, this substrate that you are able to see is now inside the what? Inside the enzyme. So, and should be able to fit perfectly well. We are going to look at that characteristic of an enzyme. Okay, so we have an enzyme, and labeling on this part of an enzyme is the active site on this opening part. This is the substrate that an enzyme is going to act on. And in this case, it has to bind to the active site, resulting into the products. But before then, there's this part where it is bound to the enzyme. And this is simply known as enzyme substrate complex. Enzyme substrate complex. So all these we should be able to understand. And these diagrams, by the way, they are just extracted from the past papers. So this is something that you need to understand. All right, let us look at enzyme action. We continue with enzyme action. One more thing that we need to understand is enzyme substrate complex. Like I've already explained on this part. So enzyme substrate complex is formed when the enzyme is bound, when the substrate is bound to the enzyme. So in the first part, we have the substrate. Okay, we have the substrate, and now we have the enzyme at the bottom. So when it fits there, this is called enzyme substrate. This is known as enzyme substrate complex. Okay, then now at the end, the substrate comes out as products. So what is formed now are products different from the substrate. So the enzyme, as you can see, it remains the same shape and all that, ready to act on the another substrate of the same type. So the enzyme remain unchanged, and that is what we mean. So the enzyme remain unchanged, but the substrate is broken down. As we can see, it was two pieces, but now we have separated. Okay. Now let us look at the enzyme specificity. So to look at enzyme specificity, what does it simply mean? So enzyme specificity, we normally look at the lock and key model. So for the lock and key model, the simple explanation of the lock and key model is where you have the enzyme as the lock and now the substrate as the key. Let us say this is the lock, then of course this is the, let me put the substrate. So in other words, the lock and key model, the enzyme is the one that acts as a lock. Then, of course, the key is a substrate. So there's a specific, as you know already, that there's a specific key for every lock. You cannot use one key to open all types of lock. I guess you understand that. So that is the way it is even when it comes to the enzyme. So this is very important to understand it. So in this case, we are saying enzyme are specific to their substrate, which means they can only act on one type of substrate. Okay, if it's an enzyme that breaks down fats, it cannot break down protein, it cannot break down carbohydrate, specifically for lipids, okay, or specifically for fats. Okay, so the shape of, again, the other thing that you need to understand about specificity is that the shape of the active site, so which means this shape of the active site of an enzyme and the substrate are complementary. Okay, complementary, it means these are able to fit, they are of the same shape with the active site. For example, you cannot bring in the substrate that looks like this and be able to fit on that part. It cannot fit on the active site, so they have to be complementary. So the keyword here is they need to be complementary. Active site of the enzyme and substrate need to be complementary. So very important that you understand this. Okay, let us look at how the enzyme works. So for the enzyme, the way it works, first thing first, the substrate binds to the active site, and of course they should be complementary in terms of shape. You understand that from where we are coming from. This is the active site. So the substrate entering the active site should be a perfect fit. Then now when it fits there, a reaction starts. This result into, or when it fits there, it results into the formation of enzyme substrate complex. So this is enzyme substrate complex. Then afterward, that is when the reaction occurs. So in this case, it results into enzyme product complex about to form products. Then from enzyme product complex, that's when the product are formed. As you can see from this, it has been broken down to two pieces. So these are what we call products. So products, where we are coming from, we said these are just substances that are produced after a reaction. And like we said, the enzyme remains the same and can be reused over and over. It can keep on acting on substrates, more substrate, over and over. It can be what? It can be reused. So this is simply how an enzyme works. So I guess you understand this very well. All right, let us look at the factors that affect enzyme activity. So enzyme activity are affected by factors such as the temperature. So we are going to just discuss temperature and pH in this case. So factors that affect enzyme activity only have temperature and pH. Now let us start with temperature. Now when you talk of temperature, that's a degree of hotness, just a degree of hotness or coldness of an object. But in this case, let us look at it as one of the factors that affect rate of enzyme activity. Now how does it happen? So you need to understand here is that the rate of enzyme activity increases with increase in what? In temperature. That is first thing that you need to understand. Now what is the reason behind? So the reason behind is that as the temperature increases, the molecules, that is the enzyme and the substrate. So molecules here, we are looking at enzyme and substrate. They gain kinetic energy. Very important. They gain what? Kinetic energy. So when you are talking of temperature, there's gain in kinetic energy. And what happened? They move. So please, this one is move faster, increasing the chances of successive molecular collision happening. They need this. They need to collide. That is when they can form. Actually, they need to collide with a certain amount of energy. Okay, but that is not our area of interest. So they need to collide and of course form enzyme substrate complex. Only after that when they can form products. So increase in temperature, in other words, increases their chance of colliding. And if they can form enzyme substrate complex, it's easy that they can form what? Products. So as the temperature increases, the molecules gain more kinetic energy and move faster, increasing the chance of successful molecular collision happening, and therefore the rate of reaction increases. So that is what you can talk of in terms of this part. Now, again, you need to understand that not all temperatures are favorable for the enzymes. So we can say all enzymes work well at a certain temperature. So not all temperatures are favorable for the all enzymes. Instead, they work at a certain temperature. And the temperature at which they work well is called optimum temperature. So optimum temperature is at the temperature at which the enzymes work best or has maximum activity. And the optimum temperature, like I said already, it varies or it differs from enzymes to enzymes. For example, the enzymes that you find in our human body, they work well around 35 degrees Celsius to 37 degrees Celsius. Or let me just say 37 degrees Celsius. That is the optimum temperature. So which means they cannot work well in the environment, let's say, which is less than 37 degrees Celsius, let's say 20 degrees Celsius, or beyond 37, let's say 50 degrees Celsius or 60 degrees Celsius or 80 degrees Celsius. Around that particular temperature, something happened to the enzyme, as we are going to look at this. Okay, so what happened above? So above the optimum temperature, the enzyme is denatured. So the key term to use is denatured and not killed. So we don't use the enzyme is killed, but instead we use the enzyme is denatured. Okay, so above that, the enzyme is denatured. Now I'm going to explain enzyme denaturation, or let me just explain before we proceed to the other part. So let us say we have this as the enzyme. So what I'm more concerned about here is the active site. As you can see, the active site, let me say this is the substrate that is able to fit on this active site. As you can see, they are complementary. But when the temperature increases now, these same enzymes, the active site is now denatured. Look at this. Let us say the active site, this part now has changed the shape. From this, it has transformed to this. Now, answer this question. Can this substrate that we have here, this substrate, still fit on that part? No, because they are not of the same shape. So enzyme being denatured, it means the active site is distorted or destroyed, and therefore the active the substrate can no longer fit. And if it can no longer fit, the rate of enzyme activity start decreasing. So therefore, high temperature denatures the enzyme and slows down the activity of the enzyme. Now, what about at low temperature? At low temperature, the enzyme remains the same, the active site and all that remains the same, and also the shape of an enzyme. But at this point, the enzyme is inactive. So sometimes you find this question. So at low temperature, we don't use the term denaturation, or enzyme is not denatured by low temperature, but instead the enzyme remains inactive. That is what slows down the rate of metabolic reaction. But as the temperature increases, the activity tend to increase until it reaches the optimum temperature. So it increases that way. So now, I think this is what I've explained. Enzyme denaturation disrupts the active site of an enzyme, making it difficult for the active, making it difficult for the substrate to fit on the active site. So whatever is here, it has to be, it disrupts the active site, making it difficult for the enzyme to fit, for the substrate to fit, okay, to the active site. So this is supposed to be the substrate to fit to the active site. I guess we've gotten something up to this point. All right. So graphically, it can be presented like this, okay? So starting, sometimes the graph we find, commonly graph are found, the ones that start from origin. So what is the interpretation of this? So at low temperature here, if this is the start, temperature going this side is increasing. So from 0, 0 up to there. Let us put this one to be the 37 degrees Celsius. Okay. So at this temperature, the enzyme is what? Inactive. Then as we increase the temperature, what happened? It gains kinetic energy and increases the chance of their colliding with the substrate, resulting into formation of substrate enzyme complex. So here, molecules gain kinetic energy, resulting into the increase in activity. or rate of reaction. Then above or beyond this optimum temperature, so if this is 37 degree, beyond 37 degree, what is happening? The active site of an enzyme has been destroyed. So the substrate can no longer fit. If it can no longer fit, then reaction cannot occur. So this one tend to reduce the rate of reaction. So in this case, the enzyme is denatured. Okay. Now, at a certain point, maybe, anyway, let me not go there. So the enzyme is denatured. This side, bottom, the enzyme is inactive. This is the optimum pH. So very important that you know this graph. Let us turn our attention to pH now as one of the factors that affect enzyme activity. Now, when you talk of pH, this is what you don't look at. So we don't talk of kinetic energy when you talk of pH. We don't talk of collision or the molecules colliding. So we don't talk of collision when you talk of pH. So pH has to do with just a condition where is it acidic or basic? Is it in acidic or basic environment? In other words, we are just looking at whether the reaction is occurring in the acidic environment or in the basic environment. Okay, so pH affects the way an enzyme works. An enzyme, or each of the enzyme, has the pH at which it works well, and that is what we call optimum pH. So just the pH at which the enzyme works the best. Now, just like temperature, below the optimum pH of an enzyme actually is inactive. Okay, so below the pH, an enzyme is inactive, and above the pH, an enzyme is denatured. So it works well within the optimum pH. Okay, graphically can be presented like this, and here we give 3 examples: pepsin, salivary amylase, pancreatic amylase. So, pancreatic lipase. So you find pepsin in the stomach. So this is in the stomach. Then of course amylase in the mouth. Then you find this in the small intestine. Okay, you find this in the small intestine. Now, these enzymes work well at different pH. For example, the pH of the stomach is approximated to be 2 pH. Then, of course, the mouth is slightly alkaline, let's say 7.2, around 7.2 there. Then, of course, the duodenum also is alkaline, let us say 8 up there. So now, these enzymes work well at different pH. For example, the enzyme in the stomach work well at acidic pH. Okay, below that pH, the enzyme is inactive, but above this pH, the enzyme is denatured. So in other words, the enzymes that work in the stomach, it cannot work in the mouth and also cannot work in the small intestine, okay, because of different in pH. So that is how you can look at in terms of pH. So increase in pH increases the rate of reaction until at a certain point called the optimum pH. At that particular point, the enzyme has maximum activity, but beyond that, the enzyme is denatured. So very important that you understand this.